Hello, everyone who is already there, and welcome to the Puro webinar on a new methodology. Let's wait a couple of minutes for people to join, and then we start the webinar. Hi. So let's give it a minute for people to enter. All right, so it's 1 minute past. Welcome everybody to this Puro Earth webinar on microalgae, carbon fixation and sinking. We have a good crowd here, so good morning, good evening, good day. My name is Marianne Tikkanen. I'm the head of standard in Puro Earth, and I will be your host today. Next, let's go first through some practicalities. So for questions, we kindly ask you to use the Q&A button at the bottom of your Zoom page and write your questions there. And then we try to answer the questions after each section and in the end of the webinar. The webinar is being recorded and that will be shared with you later. And we will send you a feedback form for the webinar, but the main thing is to get comments on the methodology. And here is the link to how to do it and how to get the document to be consulted on and the form where you can then provide your feedback to contact@puro.earth. Next, please. So this is the 8th methodology in the Puro standard. So the bottom row is where we started with the methodologies onshore or terrestrial, so to say. And now lately, during the last year, we have then brought to market the top row, the ocean methodologies, focused more on the potential that ocean offers for removing carbon and sequestering carbon. And this is now the 3rd ocean methodology that we kindly ask you to do review and give feedback in this draft version. Microalgae, carbon fixation and sinking. We can take the next. And like every methodology in the Puro standard, also this one, microalgae, carbon fixation and sinking has 2 key focus areas. One is the safety and the other one is the durable storage. So this methodology sets requirements for the projects, how to reach the durable storage of the sequestered carbon, how to make it safe for people and environment to do this activity, what has to be done to be able to quantify the carbon removal, the gross and the net. The methodology sets eligibility criteria, where and who and how can this be done, and also criteria for the risk mitigation. And a lot of monitoring has to happen to demonstrate all that. Monitoring takes place in laboratory but also models and field-based monitoring are required to be done. And all projects do have to bring their project-specific data and take the measurements from their specific site and location of deployment of this activity. But these are the very kind of general key principles of this and other methodologies. And with me here, I have the experts who have been working on this from Puro's side, also from Oceanid CCS side. And this, as we have informed, is a methodology where GigaBlue has been the initiator, and they have contributed to developing this methodology with the team. So you will now hear from the experts more details to all of these sections. Yes, and important date, we'll come back to this in the end of the webinar, is July 2nd. So you will then, before that deadline, you are asked to give your feedback to us so that we can improve. So this is still in a draft version to be approved, and your feedback is very welcome and valuable. And I know that there is expertise out there. I was surprised by how many people were interested in this. I thought that ocean methodologies are marginal, but they are emerging with over 200 people interested. So we are very lucky to be able then to get your feedback and improve. But then let's jump to the details and hear from Sebastian Haas next.
Yeah, thank you, Marianne. Trying to find my laser pointer. Yeah, let's jump into the introduction to the methodology and the technology behind microalgae carbon fixation and sinking. So I want to start out with this image of microalgae and make the point that they need this range of nutrients to grow. And while they need some of them in higher quantities than others, so they need, for example, nitrogen and phosphorus in higher quantities than, for example, iron and manganese, the absence of something like manganese would still limit their growth. And in fact, there are areas of the ocean that are called HNLC regions. HNLC stands for high nutrient, low chlorophyll. So they are high in some of these nutrients, but low in chlorophyll because they are limited, because microalgae growth in them is limited by some of these other nutrients usually. And that's an important piece of background to understand the idea for this technology. The other important aspect on the engineering side is the engineered substrate. And here we're talking about small particles that are made of organic material and that can hold nutrients. And one important feature of them is that they can float near the surface of the ocean and for specific periods of time that can be engineered basically into their material makeup. So something like, for example, a couple of weeks, they can float in the ocean and offer the nutrients that are added to them to the local microalgae. And because the local microalgae, because we're targeting areas of the ocean where the local microalgae are limited by the nutrients that are offered by the substrate, they are expected to be recruited onto the substrate, to enter the substrate and find favorable growth conditions and grow at high rates on these substrates using the offered nutrients. After this fixation phase, the density of the substrate will change and increase, which will lead to the export phase, which will lead to the sinking of the substrate along with the microalgae that has grown on them, exporting the biomass containing carbon fixed by photosynthesis into the deep ocean where it can stay for long-term sequestration. So to zoom out from that a little bit, here's an overview of the activity of microalgae carbon fixation and sinking, MCFS. So for the substrate, needs to be processed on land first, of course, and then transported out to the sea, where we can start with this deployment process that we described in the previous slide, flotation, sinking, and storage in the deep ocean. So what we're looking at here is storage in the deep ocean with its long ventilation timescales due to these global circulation patterns that keep deep ocean water away from the atmosphere on this timescale of at least hundreds of years, which leads to safe and durable carbon removal with a durability timestamp of 200 plus years. And this is done within national maritime jurisdictions. And there's a variety of strict requirements for monitoring, environmental and social safeguards, as well as carbon accounting that the methodology that we developed prescribes. And a lot of this webinar presentation will be spent on going into details on that. Before we do that, let me give you some more idea of how this technology harnesses or makes use of natural ocean mechanisms to enhance carbon dioxide uptake. So the first mechanism, natural mechanism it makes use of is the capture of CO2, carbon dioxide, by microalgae near the surface. And that's like I described, by recruiting the microalgae onto the substrate with the favorable growth conditions due to the added nutrients. So the microalgae will capture CO2 by photosynthesis and thereby deplete the near surface water of CO2. And then the second mechanism will come in, which is air-sea gas exchange. So a CO2 depleted surface water will lead to flux, to basically the water pulling CO2 from the atmosphere into the ocean because of this difference in concentration, or this gradient, as we say, between the atmosphere and the ocean, which wants to deliver CO2 into the now CO2-rich surface ocean. Now, to make this sustainable, the next natural ocean mechanism that needs to be employed by this technology is export, or also the biological carbon pump of exporting carbon that has been captured by photosynthesis near the surface water into the deep ocean. Again, that's a natural process in the ocean that is enhanced by the technology, specifically the increased density of the substrate during the export phase that allows it to sink through the ocean water column at fairly high speeds, and delivering it to the deep ocean, where we are making use, where this technology is making use of the long potential storage time of deep ocean water. So the long time that deep ocean water is isolated from the atmosphere and everything that is within it, along with it. Okay, with that overview, I'm going to go into the environmental safeguards and risks that we addressed in the methodology. So the first consideration in terms of environmental risk is to set eligibility criteria for the substrate material. So the substrate needs to be non-toxic, non-hazardous, and it's mostly consists of organic material and also some minerals, mostly the nutrients that are added to it. There are strict limits for potential toxic elements contained, so we don't want them in there. There are size range requirements for the particles. There are minimum density requirements to ensure efficient sinking. There are, on top of that, requirements for maximum flotation period and minimum sinking speed. And the nutrients attached to the substrate need to be at very low tendency to dissolve, so they wouldn't spread out in the surface water, but attract the phytoplankton to the substrate rather than just fertilizing the surface ocean. And as far as environmental risks from this technology goes, we addressed this wide range of potential risks that is listed here that actually corresponds to subheadings in this section 4.5 of the methodology. And you're welcome to read into any of these, all of these more. I will try to pick out a few of these and explain what they are about and give a sense of the types of requirements that we are setting to avoid these risks and generally to avoid, minimize, and mitigate and also assess these risks. So starting on this first point of oxygen depletion and greenhouse gas production, what this refers to is if you have a lot of microalgae growing, as you would have in this technology, microalgae in the first place produce oxygen, but they could be, in theory, degraded by microorganisms that use up oxygen, and that can cause strong decrease in oxygen under some circumstances. And that's something we are requiring to be monitored by oxygen concentration measurements throughout the water column at the deployment site, modeling of oxygen concentrations in the deep water, and generally speaking, that would inform the requirement to not decrease oxygen concentrations below a certain threshold as a consequence of this technology. And what this wants to avoid is just oxygen depletion in and of itself, being an ecological harm potentially, for example, to fish who need oxygen, and also avoid things like greenhouse gas production through certain biogeochemical pathways that open up at low oxygen concentrations that can produce greenhouse gases like methane or nitrous oxide. Next, we also addressed a bunch of other chemistry-related environmental risks. Some of these have the good potential to actually be co-benefits of this technology. For example, changes in carbon chemistry would mostly be expected to slightly raise the pH, which could counteract ocean acidification, and that would be caused by the photosynthesis. Dimethyl sulfide production could, dimethyl sulfide is is a chemical that in the atmosphere that is produced by phytoplankton, by microalgae, that can in the atmosphere favor cloud formation, which would be, climate positive, i.e. good for against climate change. And some of these are potentially core benefits, yet we are requiring to basically keep an eye on them so they don't get out of range. For example, the pH increase isn't excessive to the degree that it's actually on a short time scale with a high pH change affects ecology. So that just as an overview. Then maybe another group here is changes in biodiversity with the almost worst case scenarios, at least in the subsection of a harmful algae bloom. So for this, we've put requirements into place to monitor the microbial biodiversity at the deployment site using environmental DNA techniques like 16S RNA gene sequencing, 18S RNA sequencing to take stock and to monitor the microbial communities in the place. And again, there are requirements to limit the short-term changes in these communities. As far as larger organisms go, thinking of fish, here, as when it comes to organisms that are large enough to actually ingest the substrate, so larger fish, the substrate should just pass through their digestion tract, basically. And the most important requirement in my mind for this part, we've already addressed in the previous slide about the substrates to be non-toxic. So that's an important requirement that should also avoid harm to larger organisms that might ingest the substrate. Another aspect of this is the fast sinking speed, where there's hopefully little time for fish to ingest them. And then also there's requirements around keeping an eye by monitoring the situation and using information from fisheries reports and scientific literature to understand the risks around this for local macrobiologically communities. Okay, finally, the last point here is addressing, it's called nutrient robbing and phytoplankton growth rate changes. What we're addressing here again is sort of minimizing changes in the ecology and the biogeochemistry of the local environment, and also of the environment that might be affected by these changes downstream. And by downstream, I mean ocean currents, basically, that might carry the water in which we are growing the microalgae on the substrate now, but ocean currents might later transport that somewhere else. So there's also requirements that cover this as part of this framework that requires close monitoring by measuring nutrient concentration before, during, and after the deployment, as well as modeling phytoplankton growth rates using that are based on these nutrient measurements, as well as based on kinetic monotype models for phytoplankton growth. And then also using stoichiometric concepts like the Redfield ratio to translate nutrient concentrations into biomass and growth rates. So with all of these details, I want to end by giving a general principle that we try to follow in defining these environmental risks, and building blocks of these general principles. Of this general principle are the adherence to local permits and the adherence to local regulation and acquiring of local permits that are informed and will inform pre-operational assessment, which in turn will inform operational design. And the operational design includes measurements and modeling, as I've described for several of these examples, that are supposed to minimize and mitigate these risks, as well as inform research and, more generally speaking, our understanding of what happens in the ocean when deploying this technology. And the point I want to make here is that this is very early days for this technology. It will be commercially deployed for the first time, hopefully later this year, and it will have to grow quite a bit beyond that, and scale quite a bit beyond that. So especially now putting into place some of these measurements and modeling requirements that might address risks that at this relatively small scales are probably many of them quite low. That can help us to understand for when this technology scales to larger levels, how to do responsible pre-operational assessment and operational design in the future when this scales. Okay, and that will minimize and mitigate those risks, hopefully in the long term. And that's it for my environmental risk section. And I believe before we go to the next section, we might have a few questions.
Yeah, let's see if we have something here. Thank you, Sebastian. Let's take a couple of questions here. So keep your questions coming in the Q&A button, and we'll then take more of those at the end. Sebastian, there was a question about what type of waters does this methodology work? Can it be applied to also seas, rivers, lakes, any water type?
Okay. Yeah, so currently in its current form, it's restricted to the ocean and even restricted to these HNLC regions of the ocean, the high nutrient low chlorophyll regions. So that would have to be a separate methodology, in my understanding, to allow the deployment outside of these areas.
Okay, thank you. Let's leave it at that. I'll take more questions in the end. In interest of time, let's hand over to Ben, Michaela, and Kana on the quantification. And
Great.
Next slide, please. Sebastian, can you? Thank you.
There we are. Thank you, Marianne. Hi, everybody. So this next section we'll be talking about the carbon accounting that goes into this methodology and the cork calculation and how the carbon credits are issued and calculated. And so the cork standard is pretty standard across the different URO standards, and we'll be going into different components of each. And so for this particular methodology, our C stored will be our gross carbon removal, our gross carbon dioxide removal credits. And in this case, it's calculated by we deployed the substrate, it's floating on the surface, we're accumulating the microalgae and the biomass among the substrate, and then it's going to sink to depths, and then creating this CO2 deficit within the upper waters, which will be replaced from the atmosphere. And so this will be kind of our core carbon accounting methods, and we'll account for how much has been stored, how much has reached the depth, and how much has been replaced from the atmosphere. And kind of durably stored. And then so subtracting from that will be a number of different factors that kind of subtracted from this gross total of carbon sequestered. And so the first of those will be the baseline measurement. And so the baseline reflects what would have happened without this project intervention. And there are two key components to this baseline estimation. One is how much biological production would have occurred within the project area if the project had not occurred, how much microalgae would have grown or other life would have grown kind of without the project, and that the project developer doesn't deserve credit for. And then the second critical piece of the baseline assessment is part of this methodology is that we are delivering nutrients which are stimulating biological production, but there'll be some nutrients that'll be naturally present that are not being introduced. And so those nutrients will also be delivered to durable storage. But they may be kind of nutrient robbing, and so maybe a long time later or at a distance, biological production that would have taken place without this introduction, would now not be taking place, and so that would also be counted against the carbon accounting. Now, in this case, it is from a carbon accounting perspective, we really are interested only in biological production in both these phases that would have resulted in durable storage. From an ecosystem assessment perspective, we care about any changes in biological production. But from a carbon accounting perspective, in terms of the baseline assessment, we're caring about biological production that would have naturally occurred, which would also have resulted in durable carbon storage. And that's our baseline. For our C loss calculation, so this is carbon that has reached depth that has been stored, but then is going to kind of be released into the atmosphere before the end of the credit period. So this standard will have a 200-year plus durability standard. So we're saying that any of the carbon that's being sequestered by this method must remain sequestered, must remove from the atmosphere for at least 200 years. And so in this case, the carbon's been delivered to the ocean depths. It is our expectation that given the multiple century time frame that we're looking at, that carbon will decompose, that will dissolve. And so there's a risk that it will be kind of upwelled, reach the surface, and then be re-released into the atmosphere. And so the main component of this carbon loss segment is how much of that carbon is going to be re-released into the atmosphere. And then the next component that will detract from the total cork calculation is this emissions for your project. These could be any sort of emissions that are part of any phase of the project. So it could be during the production of the substrate, could be during transportation. It could include indirect emissions such as power drawn from the energy grid. And so any of these emissions that are related to the project itself are deducted from the cork total. And then the last piece of the calculation is the emission leakage or other kind of leakage. And these would be kind of other activities that may have taken place if this project had not gone forward. And so there'd be other kind of negative ecological things or other kind of project shifts, market shifts that would be. And so less relevant perhaps for this particular specification, but still want to be kind of accounted for within the formula. And so each of these parameters has its own kind of tricks and complexities, so specifically for this particular standard, this particular methodology. And so the next couple of slides, we'll be going through kind of how we're calculating some of the aspects of these different calculations. And so for that, I will turn it over to my colleague, Michaela Sten.
Thank you, Ben. As Ben introduced, carbon stored is the amount of carbon dioxide that is durably stored for longer than 200 years. And the way that the methodology currently quantifies that is it is a function of the carbon that is affixed to the substrate that's deployed, and the amount of carbon that's able to make it from the surface ocean to the seafloor, and the fraction of the air-sea gas exchange that occurs at the surface. Next slide. And the way that this works in practice is that during the initial release of the substrate, there's a carbon fixation phase in which phytoplankton grow on the substrate. And the biomass of this substrate is considered the carbon that will eventually be stored, and that is part of the sea fixed. Once the substrate enters a sinking stage or this export phase, that carbon on the substrate is then moved from the surface ocean to the seafloor. And because phytoplankton are a relatively easy to eat by microbes material, during the sinking phase, some of that carbon falls off of the substrate during the sinking process through a variety of microbial processes. And so the sinking efficiency measures how much carbon makes it from the surface ocean all the way to the seafloor, at which it is then able to be durably stored for at least 200 years. And then finally, because there is inefficiency between how much the ocean is able to absorb carbon dioxide from the atmosphere, there is a air-sea gas exchange fraction, which determines how much of the carbon that is moved from the surface ocean to the seafloor is then able to be taken up from the surface, from the atmosphere into the surface ocean. And the caveats that I will include to this is that the biomass we only count the biomass that's affixed to the substrate, and any biomass that is not affixed to the substrate during this process is not included in this cork. Next slide, please. And so the quantification of the carbon fixed is all of that biomass that accumulates on top of the substrate. And the way that this is quantified is the substrate is measured pre and post deployment to account for the substrate weight itself, as well as the biomass that is affixed to the substrate. We then require quantification of the percent of substrate that sinks out of the surface ocean and makes it to the seafloor. We then have quantification requirements for the amount of biomass and the type of biomass that is affixed to the substrate in order to accurately quantify the carbon dioxide that is being pulled out of the system. And then we recommend a conversion factor for carbon into carbon dioxide so that we can accurately quantify the carbon dioxide that's being pulled out of the system. Next slide.
Okay, so jumping in here, I'm gonna cover the air-sea gas exchange. As Michaela really well described, this marine microalgae carbon fixation and sinking is going to remove carbon from the surface ocean, which is quantified by the carbon fixed term and the sinking efficiency. But we also need to examine this air-sea gas exchange term to quantify how much of the carbon is removed from the atmosphere and taken up into the ocean. So this AS term is going to be both a factor of the carbon dioxide gas exchange rate, as well as the movement of those surface water masses. And because of that, the complete equilibration of the ocean and atmosphere CO2 can take many years to even centuries. So to quantify this, we need to use models. And here in this case, Perot is proposing the use of the direct ocean removal tool created by Carbon Plan and Seaworthy. This is publicly available and online. And we acknowledge that this direct ocean removal, DOR tool, is specifically for the abiotic removal of carbon from the surface ocean, and therefore was not necessarily designed for biological pathways such as this one here. This means that biological feedbacks, like changes to alkalinity and nutrient cycling, are not accounted for in this data set. So to be clear, we are using this tool solely to quantify this gas exchange efficiency as a result of this carbon removal from MCFS. So there are several different sort of settings on this tool. We're proposing that we take this efficiency metric at 10 years for a specific region that overlaps with the deployment site. Next slide. And then the last term, which is a reduction term in the Cork evaluation that Ben had introduced, is this carbon loss term. So with any biological pathway in the ocean, we need to consider the degradation of that carbon as it reaches, as it's stored at the seafloor. And as that carbon degrades, it can be sort of recirculated and recycled back into the atmosphere. So that's going to be this loss term. And we calculate this based on the amount of carbon that makes it to durable storage, which is going to be that C fixed and the carbon fixed and the sinking efficiency product as well. And we add this third term, which is this fraction ventilated. Now, in this case, because the permanence, the permanence year is a minimum of 200 years, the fraction ventilated is going to be the amount of carbon that has been sort of mixed out and re-emitted back into the atmosphere within those 200 years before your durability. Next slide, please. So again, this requires the use of models because these will entail global circulation dynamics. And here I'm showing you what you might expect from a model results that shows you the fraction of dissolved inorganic carbon that's remaining 200 years after it's been deposited at the seafloor. So this is data from a published work, Noik et al. 2024, and we can see that in many parts of the ocean, within 200 years, a majority of that carbon that you've stored at the seafloor is preserved, but in certain places, in the sort of orange and yellow to green areas, we're losing something like, anywhere from 20 to 40, 60% of that carbon within those 200 years. So that will essentially get deducted from your net carbon removal. And I think this is the last slide for the carbon quantification. So I just wanted to really reiterate that we would very much appreciate your feedback on all of the details in the methodology. And we very much welcome any ways to improve it.
Thank you, Kana and Kaylee, Michaela and Ben. And thank you for the questions. There are questions that we can now take time to answer, at least some of them here, and I may be paraphrasing or combining the questions here. So Sebastian, I think there were questions about which might fall into your area of expertise about how widely does the falling microalgae spread, and are there any worries about smothering it at the sea bottom?
Mm-hmm. Yeah, so we're expecting we would be expecting the substrate plume to be spreading pretty widely on its long way down through the water column and due to ocean currents. There are requirements to actually ensure that, or to monitor whether that's actually the case, for several reasons. Specifically, this aspect of smothering of benthic organisms, and what we mean by that is benthic organisms means organisms living on the seafloor, things like sea cucumbers, sea stars, and the risk here, the environmental risk being addressed here is the too much covering by a lot of substrate falling into a restricted area of the seafloor, and the main thing this is addressed is by monitoring of the bloom, to what extent it spreads out. And additionally, there's requirements around pre-assessment of benthic ecosystems and organisms in the deployment area to assess that beforehand, whether there's any sensitive organisms.
Thank you. Then there are a couple of questions that are about the fractions, how much is trapped in the sediment and how much is assumed not to be in the sediment but more elsewhere in the waters, and how is that quantified? Who can take that question?
Take this. So we assume that once the biomass reaches the seafloor, because we don't have a good way of quantifying the incorporation of that biomass into the sediment, that is all immediately mobilized into DIC and then can move throughout the ocean system. However, there are some monitoring constraints to make sure that if it is incorporated into the sediment, that we can minimize things like smothering and we can monitor for things like that. But in terms of the corks, we assume that it is all converted immediately into DIC, which is a conservative estimate for these timescales.
Thank you, Michaela. And maybe let's continue on that path. There are several questions on the instruments and the equipment that are needed. Also a question about does this need sampling from the seafloor? So if you can generally speak to what kind of instruments are foreseen to be used here.
I definitely invite everybody who are curious about those specifics to definitely dig into the methodology in section 9, where a lot of this is described. I will say there are basically a mix of pre and during and post-deployment monitoring that includes sampling of the deployed substances, in situ monitoring of tracking the particles, their sinking speeds, doing lab analysis on the substrates after, before and after it's been deployed, and of course a lot of modeling work going into the long-term impacts.
And the seafloor sampling, is that a must or not?
Actually, I'm wondering if Sebastian, because seafloor sampling is probably more directly tied to the environmental risks, if you want to touch on that.
So in terms of environmental risks, there's no seafloor sampling. So a few areas touched by that is bottom water oxygen, which we are monitoring using via the water column oxygen concentrations, as well as box models for the bottom water to understand the development of oxygen concentrations near the sediment. But for the environmental risk sections, there's no requirements that that would necessitate sediment sampling. And so for the quantification part, is there a question, Marianne, for sediment sampling for the quantification part? My understanding is, without being intimately involved in that part of the methodology, is that the particle tracking mostly occurs through these Lagrangian type particle tracking models that replace like direct observations, which seem difficult to do. And please correct me if I'm wrong here. My understanding is that it seems just not really realistic to expect to find the right places in the bottom of the ocean, because to actually sediment sample, since the substrate is expected to be spread out far and wide. So this is solved by modeling, in my understanding.
Okay, thank you. And in interest of time, I would actually then maybe finish off with the final question, and that is on the durability. So there is, and maybe you addressed this in the presentation, but good to repeat. So how and why is the 200 years the baseline storage mark or durability? Who can address that?
Is that a question of determining the baseline for 200 years, or just sort of understanding, or
maybe
setting
more
that
in
200
general,
years?
where does the minimum 200 years come, and why?
Yes. Yes. So as I mentioned, with biological pathways, there's a risk of this return of carbon after it's been degraded over time and re-released back into the atmosphere. And in some places, this fraction of re-release can be relatively low for several centuries, but in other cases, that's not necessarily the case. And so this kind of allows us to set a minimum range of 200 years. And you can see that, if you're in some deployment sites that have a higher risk of this re-release, then that gets deducted into your quark, while places that have much stronger durability is going to have a lower, potentially a very 1-2% deduction in those 200 years.
Okay, so minimum is really a minimum, and it can be exceeded locally in different conditions of the ocean.
Yeah.
But then I would like to actually close the webinar. We are a little bit over the 45 minutes, and Sebastian, if you can go to the slide to remind people that we are very much in the phase of developing the methodology. This is the reason for having the public consultation. It's really nice to see the crowd here that is engaged and interested in this area. So please chip in, give your views, your experience, and so that this can be evolved to a methodology that can then be used by the projects. So July 2nd is the important date. The links were also shared in the answers here. They are also here on this slide. And then we are going to address all the comments that we receive, and then make changes where we can improve. So thank you, presenters. Thank you, participants. And we hope to hear from you soon.